and give us a brief overview of what he's about to present. So, Superintendent Boampong, you may share your slides now and give us a brief overview of what he's about to present. So, Superintendent Boampong, you may kindly share your slides now. Introduce our speaker for today.
All right. Thank you very much. Good afternoon.
Give us a
good afternoon, all.
Good afternoon.
Yeah,
good afternoon, Professor.
You're welcome. Yes, you are welcome, seniors, to our monthly seminar series. Today we are fortunate to have Mr. Emmanuel Boampong to speak to us on a very important and interesting topic. But before I talk about the topic, I would like to share a brief profile of Mr. Emmanuel Boampong. Mr. Emmanuel Boampong is one of our own. When I say he's one of our own, in fact, he completed his BSc from the University of Cape Coast. He did BSc in biochemistry. Currently, he is a superintendent of police working as a forensic scientist with the National Forensic Science Laboratory of the Ghana Police Service. He also holds a Master's of Public Health from University of Ghana and a joint Master's in International Affairs and Diplomacy. He received international training in forensic science, crime scene investigation, and DNA analysis from the Lutheran Police Forensic Science Center. He was awarded an international fellowship on nuclear forensic analytical measurement by the IAEA, which was executed at the prestigious Hungarian Academy of Sciences. He is a member of the International Society for Forensic Genetics, the Biochemical Society UK, and has also been awarded a post-nominal title of member of the Royal Society of Chemistry. He represented Ghana as an expert advisor for the BWC's 8th Review Conference. And he has involved in and supervised the design and delivery of varied training for personnel. He is a certified trainer in drug analysis with the United Nations Office on Drugs and Crime, and in assistance and protection with the Organization for the Prohibition of Chemical Weapons. So indeed, the experience he has fit, our scope. And therefore, we thought it wise that we need to invite him to speak to us on a topic outlining the forensic DNA process at the Ghana Police Forensic Science Laboratory using the human identification of 3 case studies. But this topic is very, very important, and especially to our students. And therefore, we ensure that all our students join this particular forum to listen to this important topic. So, without much, I would like to invite Mr. Emmanuel Boampong to talk to us on this interesting topic. So, Mr. Boampong, you are welcome.
Hello, can you hear me?
Yeah, we can hear you.
Okay, thank you.
Yeah. So please, I hand over the podium to you to talk to us on your topic. Thank you very much.
Thank you too, Prof. Very good afternoon to Prof. That's the head of department and all the faculty at the Forensic Science Department. And then also a very good afternoon to all those who are joining, the students and every other person.
Yeah, Boampong.
Hello.
Can I come in just a minute? Sorry. In fact, we have an important personality joining us. We have the commandant of the academy, police academy, joining us. And so I would like to acknowledge him. DCOP, we are so grateful, sir, for joining us.
Thank you, Prof.
Yeah.
All correct, sir.
Thank you, Boampong.
All right, take over, Senior.
Yes, sir. Okay. Hello.
Yeah, we are listening to you.
Okay.
Yes.
Yeah.
A happy Eid to all the Muslims on present here in the seminar today. I would first start off by wishing all of us well, and then also zooming into what I need to present today. I'm hoping I would not take so much of the time so that we can use the remaining of the time to do some interactions, questions and answers, and then we'll be able to deal with that. So I'm counting on my moderator to alert me when I'm exceeding 45 minutes or 30 minutes. Okay.
All right.
Thank you.
All right.
I think much has been said about me and the topic that I'm going to present on. This is, I hope you all can see my screen.
Yes.
Okay.
Yes.
Can you, I think it's still not on the slideshow. Can you put it in the slideshow, please?
It's not on the slideshow.
Yes.
Oh. Okay, let me try it again. Is it better now?
Yeah, good.
Is it better now?
Yeah, that's good.
Okay. So this is the outline of my presentation. We'll first be dealing with the introductory parts, where we'll be handling some historical developments, especially for the students. And then,
Mr. Boampong.
Hello.
Yeah, can you? I had Dr. Edgar Akono. I think he has raised up his hand. I don't know if he has. Yeah.
Okay.
Dr. Akono.
Hello. I think it's okay. It's okay. We can continue, sir.
Okay. Thank you so much, sir.
All right.
You're welcome, sir.
Okay. So as I was saying earlier, we'll be dealing with some introductory parts, and then we'll be zooming into the 3 case studies that I talked, I indicated in my topic. First of all, for the historical developments with DNA analysis, especially for the students, I would date back to the 1900s. where the emergence of or the discovery of blood grouping, as in forensic hemogenetics, was established. That was really done by Landsteiner and then Leon Lattice in the 1900s. And then in the 1930s, the genotyping technique, the ABO genotyping technique took grounds with the development of several serological markers and blood serum markers. In the 50s, we had two gentlemen, Watson and Crick, discovering the structure of the DNA as a double-stranded molecule with helical arrangements. And then in the 60s and 70s, we also had several other techniques coming up, the southern blotting, Sanger sequencing technique, and then restriction of enzymes coming up during that period. In the 80s, the first polymorphic locus was reported. And then in 1983, Carrie Mullis discovered a PCR technology, which obviously won her a Nobel Prize during that period. In 1984, Sir Alec Jeffreys was the first person to use DNA in criminal investigations to identify a suspect in the UK. And that basically formed the modern day evolution of DNA analysis for forensic purposes. A little skip back to the Ghana's Police Forensic Science Lab. The establishment started within the 1960s, and I'm happy the commandant is here because everything in the 60s, everything was being housed at the police training school. Then it was just the crime scene, the fingerprint, and the photography sections that were established during that period. Over the years in the 70s, there was a split, and so the crime scene and fingerprints units were taken to the place that is currently the Greater Accra Regional Command. And then the photography section was being housed at the current place where the police training school. That then it was just the crime scene, the fingerprint 1960s by the photography sections that were established during that period. In the 80s, the chemistry section or the narcotics section was added on. And then in the 90s, we had the document examination and then the biology section. Then for the biology section, it was mainly serological analysis. And so there was no DNA being done during that period. And then fast track into 2010, we had the renovation of the lab and then the addition of the DNA unit, as in the upgrade of the serological unit into the DNA analysis unit to its current status. I hope my picture came out well, but this is a photograph of our current forensic science lab. Probably might not have come out well, but then I do believe some of you have already been there, so the photograph will not be so essential for you at this moment. Now, the main sources we look at or we deal with at the forensic lab, mainly wet or dry blood, wet or dry saliva or semen, hard tissue, that is bone and teeth, nails, epithelial cells, hair with follicles, high vaginal swab, and then buccal swabs mainly for reference samples. The basic thing about DNA that we need to talk about here. The first one is that the DNA molecule is a polymer of nucleotides. That's why you have the full name being the deoxyribonucleic acid. Deoxyribose being a 5-carbon sugar, and then a phosphate group attached to the structure. And then as part of it, we have 4 bases, 2 purines and 2 pyrimidines. The purines are what we usually term as A and G, and then the pyrimidines as C and T, cytosine and there are 2 complete copies of the genome in each nucleus of a human cell. Then you're having over 3.2 billion base pairs of information within those 2 complete copies. And these are located within the 23 pairs of chromosomes, autosomal chromosomes within the human being. For the genes that we are looking at, generally genes are regions of the DNA that encode and regulate for protein synthesis. There are roughly 20 to 25,000 in the human genome. And of this, 1.5% of that encodes for protein. 23.5% are regulatory in nature. They regulate protein synthesis. And then the remaining 75% are estrogenic, as in outside. For that 75%, of that 75%, 50% of that is repetitive DNA, and the remaining portion is interspersed repeats. For the interspersed repeats, you're having short interspersed repeats, long, and then long terminal repeats, DNA transposons, of which you are having the satellite DNA, the mini satellite DNA, and then the micro satellite DNA. For some people, these might be a bit technical, but then just bear with me for now. Then the main thing that is being considered for any STR, when I say STR, that's the short tandem repeats. I've mentioned earlier the repetitive nature of DNA, and when I mention the tandem repeats, I'm usually looking at the satellite DNA, the mini satellite DNA, and then the micro satellite. So for any locus, any short tandem repeat locus to be considered viable for forensic applications, it must be highly polymorphic. It must be easy and cheap to characterize or identify. It must also be simple to interpret, and it must be easy to compare with others. So for all that I've said, this is a pictorial representation of the things I stated earlier. Now, to the steps in which we use for DNA processing, I've captured them in 5 main steps. Most of the times is the 4 main steps that will be seen. The pre-extraction stage will not be seen so much in several publications. Is the extraction, the quantification, the amplification, and then the separation and detection techniques seen. But I brought in the pre-extraction stage. I would explain later why that was brought in. Then in all these, the chain of custody procedure it's very essential in bringing a resolution to the matter that is being investigated. For the pre-extraction stage, it starts basically, if it's a criminal case, it starts basically from the crime scene. There you are dealing with the collection of exhibits, the characterization, the transport, and then the storage. And then also in the lab, you are looking at the physical examination, the serological analysis to prove whether it's human blood or whether it's semen that is present in these cases. And then the sampling comes in. Then in cases of hard tissue, as in bones and teeth, there's a portion for sample preparation. Because those are difficult to deal with, we need to prepare the sample or the tissue that has been brought to the lab into a powdery form from which extraction can take place. And so these are the 5 that are catered for in the pre-extraction stage. For the extraction stage, you're mainly looking at, having a look at the picture by the side, you're mainly looking at four sub-stages. That is the lysis stages, as in breaking it down, the binding stage, the wash, and then the elution stage. Lysine, we need, because the DNA is found within the cell, we need to break up the cell and its components to be able to release the DNA. Then we bind the DNA to substrate to prevent it from being washed away when we go into the washing substage. The washing substage is to clear every other thing aside the DNA that is of interest to us. And then from that, we, should I say, debind, or we elute that DNA into solution, and that becomes our extracted DNA. Now, for the principles or the techniques of extraction, there are over 13 of them that are in use, but the most popular ones are the ones that I've highlighted. That's the silicon column based DNA extraction method, and then the Kilex 100 extraction method. Each of them has its own advantage, especially for the silicon based method that yields, it provides a higher yield of DNA. And then for the Kilex 100 extraction method, there's less contamination with the samples that you are working with. And so you get a more pure source of DNA to work with. There are several others available depending on what kind of extraction or what sample you are working with. There are bacterial DNA extraction techniques. There are plant DNA extraction techniques. But then these are the two major ones that we mainly look at. For us, we've used just about almost everything. We've gone through almost all the techniques here. But then these are the two major ones that we mostly stick to, and also based on the reagents that we do purchase, the protocols that do come with it. This is just a table of some sample types, and then a range or an amount of available DNA within the sample that is brought. Or if you are looking at liquid blood sample, within the liquid blood sample, it's estimated that you are having about 30,000 nanograms per mil of DNA within that. So based on the extraction procedure you choose and the techniques you are using, you can extract or generate an amount within range. It may not be the exact value, it may be way less than that, but it should be within a certain range so that you can proceed from there to do your quantitation. Now for the quantification, it's basically to assess the amount or the quantity of DNA that we've extracted, and then also its quality, so that we can proceed from there to do the the amplification and all the other process that needs to proceed from there. There are basically 5 types of quantification procedures or techniques available. But then for our lab, and the most popular one that is in use, that's the real-time PCR, which targets DNA-based specific methods. And for that, we are using a multiplex real-time PCR. For that, the technique that we are using is able to target several loci at a point in time, and that's why we call it the multiplex real-time PCR. For the amplification stage, the main stages or the main sub-stages for the amplification stages, you are looking at the denaturation, the annealing, and then the final extension stages. Denaturation, you are trying to break it up, separate the double helix structure of the DNA and the 2 strands. You're trying to separate them. And then the annealing deals with bringing some complementarity in terms of the purines and then the pyrimidines, how they do match up to form a new strand along each strand. And so that is how the multiplication or the amplification takes place. And for this, it's usually done within a 28 to 32 cycle framework. But then for our place, we found it to be essential to use the 29 cycle process to get 1 billion copies of each DNA molecule. And so it's for the types or the techniques that are available, we use the PCR, that's the polymerase chain reaction. And for the, just about the main last stage, that's the separation and detection. We are, for the gold standard, or the most valuable standard that is being used globally for separation and detection is the autosomal STR profiling. And in doing so, the techniques that are available, it's mainly because the DNA is charged, and then you want to separate it on a charge to mass ratio. The main application that is available currently is electrophoresis. In other places, you can use LCMS, as in liquid chromatography coupled with mass spectrometry. But then the most ideal one that is being used is electrophoresis. And for electrophoresis, you are having either the gel electrophoresis or the capillary electrophoresis. For our place, we use the capillary electrophoresis, the 8 capillary electrophoresis, which is a more advanced form. I know that in most institutions, in schools and all that, the gel electrophoresis is being used because that one is way cheaper, minimally cost effective than the capillary electrophoresis. But that takes time. And then that one is easily used for reference samples. But then if you are dealing with crime scene cases, you need to be able to have a highly specific technique which can easily separate each of these samples and the loci that are being targeted. And so it's more ideal to go in for the capillary electrophoresis to use that. And so from there, the genotype determination is done, and then a comparison of genotypes are done with the ladder that is being used, and then also in sample, in project comparisons are done. Okay. A brief about the 3 cases that we are looking at today. For case 1, this incident happened in 2022, that's last year. A male suspect robbed a businessman, this time not at gunpoint, but at, should I say knife point? Okay, because he used the knife to the throat of the businesswoman and then robbed her. And then proceeded from that to rape her in front of her 2 daughters. After the incident, the suspect cleaned his manhood with the bedsheet and then bolted the scene. Later on, I think about a month later, the suspect was arrested, and then samples were taken for analysis. For case 2, this happened in 2017. An unknown decomposed body suspected to be the missing person was recovered from a hospital's main, who had escaped from a hospital's male ward, was identified. It was the skeletal remains that were seen there. And so they were, those were brought to the lab for comparison, for identification and then comparison. And a reference sample was picked from the deceased son, the alleged deceased son. Then for the case 3, this is a bit similar to case 2. Also, an unknown decomposed body was brought in. It was suspected to be the missing person who was involved in a mob action. He had actually been beaten up by a mob on a highway because the mob suspected that he had stolen a phone from someone within the mob who had shouted thief, thief. And so a mob action took place, and then that incident happened. For each of these three cases, the first one, a stained dress was brought in, a stained underwear. Then the bed sheet was also brought in. And then reference samples were picked from both the suspect and then the victim. For case 2, it was the fingernails and then the left femur bone, together with of the deceased person, together with the alleged son, that were brought in. And then for the case 3, it was still the femur bone, the teeth, and then the buccal swab. That's the reference sample of the alleged father. Now, for the 5 steps that I indicated earlier, the pre-extraction, the extraction, and then the quantification and amplification and separation technique stages that I listed earlier. For the pre-extraction stage, all the sub stages I listed earlier took place, except for the cases 2 and 3, which involved hard tissue. That's bone, some bone and teeth samples. And so for those ones, we needed to prepare that into powdery form before we could proceed into the extraction stage. In extraction, we use the DNA investigator kit. And then based on the validation techniques and the protocol that were provided by the manufacturer and how best is suited our lab, we use that protocol for the extraction. Same was done for quantification, where we had to use the 7500 real-time PCR as the equipment, and then we use the quantifier trio kit from Applied Biosystems. Majority of our kits that we use in our lab, and then some of the equipments that we use are from Applied Biosystems or Thermo Fisher Scientific, if I should put it that way. Generally, they have the monopoly globally, so majority of the staff have to come from them. Multiplexing, as in amplification, we had to use the Global Filer PCR amplification kit. And then for the separation and detection techniques, as I mentioned earlier, we use the 3500 genetic analyzer to perform electrophoresis on the amplified STR targets, and analysis and profiling were done with the GMapper IDX version 1.5 software. Now, this is the quantitation kit. That's the DNA investigator kit. This is also a profiler kit, which is mainly used for bone samples, which if you want to extract bone samples, it's ideal to use the profiler BTA kit. Then the seminal fluid kit, that's from Seratech, is used mainly for serology to identify or to characterize suspected semen samples. And then we have the quantifier trio kit on the top right, and the Global Filer PCR amplification kit on the down left. For this mortar and pestle, I think most of you are familiar with it, and especially the students in the university. It's generally a crude method at this moment, but if you want to pulverize, or if you want to make bone into a powdery form, bone and teeth into a powdery form. This, coupled with liquid nitrogen, is used for that purpose. You cut the bone into smaller pieces, and then you put them in this, you add liquid nitrogen gradually, you grind, you add, you grind, and in that sense, till you get it in a powdery form. And that's for some of the cases which involve bone and teeth sample. Those are some of the methods we use. It's the same method, or it's the same procedure we used for the Takrady case. The lab has been involved in some high-profile cases over the years, Yana's case and several other cases over the years. But I don't want to touch on those ones. Okay, so on the left is the real-time PCR system with its computer, where you'll be able to monitor the process as it goes on and then be able to see how well your samples are being amplified. To the top right is the GenAmp PCR system, which is a thermal cycler. It's mainly used for the STR amplification process. And then down right is the ABI 3500 genetic analyzer, which we use for separation. It's mainly the capillary electrophoresis equipment which we use for separation and detection. For our results and finding, for case 1, these are preliminary. For case 1, exhibit A tested negative to the presence of human semen. That was, and then exhibit B tested positive to the presence of human semen. Exhibit B was the bed sheet that the suspect used to wipe his genitals. And C, same. For case 2, both exhibits were determined to be single source profiles, and then same for case 3. These were the concentrations and the internal PCR controls, that the values for these that were generated for each of the samples. For case 1, for the dress, it was 0.3 nanograms per microliters. And bear in mind that the real-time PCR technique is able to detect concentrations way lower than this. And then the electrophoresis process is able to work with such concentrations. And so they are more ideal for crime scene cases where you are not likely to get cells or DNA samples in higher concentrations like that of reference samples. B was the underwear, which generated 0.215 nanogram per microliters. C was the bed sheets, 0.339. And then D was the suspect's sample, E was the victim samples. And so if you look at these two, the suspect and the victim samples, and compare them to those retrieved from the crime scene, you could see a very high concentrations for the suspect and the victim, because those are reference samples. Those are from a known source, and they've not been exposed to the environmental conditions that the crime scene samples have been exposed to. The same was for case 2, the deceased sample, that's the bone that was recovered, or the skeletal remain that was brought to the lab, generated 0.261 nanogram per microliters. And then the Alessan, that was 13.4, similar to a reference sample that has been extracted. For case 1, these are the profiles that were generated. Now, if you look at these, for the students, these are alleles. The numbers that you see here are alleles. These, in other sense, these are the number of times the STR target or the STR sequence was repeated, if I should put it that way. And so for each of these loci, there are two alleles, and for an allele is an alternate of the original one. So there are two alleles that are generated for each allele, for each locus. For the victim, and especially since these are crime scene samples being compared to reference samples, it's assumed that it's the same person that left his sample or left his trace, as in Locard's principle, where every contact leaves a trace. When the suspect was at the scene, he left a trace, which we are using that trace to get back to him. And so those from the crime scene, we are looking at that, and then we are comparing that to either the victim or the suspect to be able to determine who and who contributed to the crime scene samples. If there's nothing of the suspect within the crime scene samples, then the suspect is free to go, and the investigators would have to search for a different suspect. Now, since this is a direct comparison, it's not like in the case of a paternity or relationship testing. Since this is a direct comparison, we are expecting that the profiles of the suspects, all the alleles that we see for the suspect at each and every locus, should match to whichever crime scene sample that you are testing it against, or you are comparing it against. Similarly, for the victim, we usually pick that of the victim, one, in cases of exclusion, and then two, in incidents where for the crime scene sample, there's a mixture of profiles. We'll use that of the victim to extract or to take out what is not the suspect, not of the suspect. And then we would have a clear profile of what comes from the suspect to be able to compare it to that of the suspect. And so in most times, especially with the high vaginal swabs or rape cases, differential extractions are performed to be able to separate the victim's profile from the suspect's profile. And there are instances you can have more than one suspect. If, let's say, a rape case, you can have more than one suspect or more than one person raping the victim. And so in the profile, the mixture of profiles, you are going to see that in the mixture of profiles. And so having a look at the suspect's profile and that of the victim's profile, it can be deduced that the victim's profile is directly matching that of Exhibit A, which is not surprising because that is a dress from the victim herself. Initially, the investigators suspected that there might be some stains on it, but those stains were not, one, they were not semen, and two, they were not from any of the persons that were brought in. But then the body cells that were left on the dress was what was profiled here. And so if you look at all the locus, all the loci that are seen here, between the victim and then the dress, that's Exhibit A, you will be able to tell that this is a direct match. So the victim is the one that contributed to is the only source for the dress, as in Exhibit A. Now, because it's a criminal case, let's focus on the suspect now. If we are comparing the suspect to Exhibit A, we see that there's nothing matching between the suspect and then the dress, as in Exhibit A. And so the suspect is excluded from Exhibit A. But then skipping to Exhibit C, if you look at the profiles for these two, the Exhibit C and then the suspect, is a direct match to the suspect. And so the suspect is the only contributor to Exhibit C. That's the best sheet, which he used to write himself after the act. Looking at Exhibit B, now you're having more than 2 alleles at each of these locus. And so that tells you that this is a mixture of profiles. Now, in our case, what we did was we had to extract, because we had extracted the victim's profile separately and then the suspect's profile separately, we're able to generate who and who contributed to this underwear. And it was determined as such. I think I have some of the electropherograms here. I hope I can show something here before I move on. Okay, so these are the electropherograms that were generated. It's from this that we did the table that is I just showed you. So if you look up, you can see the loci that I indicated earlier. Each of these loci are indicated at the top here, and then the electropherograms, the peaks are shown for each of these places, and then the respective alleles are called for each of these loci. So if you can see it from here, at D3, you are having, it only gave you 17 here, and it's an indication here that this is homozygous, as in a repetitive format. So it will be 1717. If you're looking down here, it's only 17 that is written there. The machine called it 17, but then you, the analyst, should know that this is a homozygous sample, and so it's a 1717. Then for the next one is same 16, so it's also homozygous, so 1616. And then for D16, at that it's an indication here, it's heterozygous. This is homozygous 13, so you're seeing as in a repetitive format, so it will be 1717. It follows. If you're looking down here, it's only 17 that is written there. The machine code is 17, but then you, the analyst, should know that this is a homozygous sample, and so it's a 1717. And then we do some allele frequency. Then for the next one is same 16. And then so it's also homozygous, so 1616. And then for the work that D16, at that it's so that was a sample, that was one of the reference samples in that order. For Q2, this was even though it was a crime scene, it was a criminal case, in one way or the other, we had to treat it as 1717. Then we do some allele frequency. Then for the next one is same 16. Also, and so it's also homozygous, so 1616. And then for the work that D16, we had to treat it that way. So that was a sample. That was one of the reference samples. It was suspected to be in that order. And a relative of that person was available. This was in this case, that was even though it was a crime scene, the alleged son, it was a criminal case. In this instance, in one way or the other, we had to treat it as 1717. And then we do some allele frequency. And then for the next one is same 16. Then we refer to that as paternity. If a mother is the one that is in doubt, we refer to that as maternity. We've had each of these instances happening at our end. And so for relationship testing instances, And we don't deal with that person was available. This was a case that was, even though it was a crime scene alleged, it was a criminal case. In this instance, in one way or the other, we had to treat it as 1717. Then we do some allele sweeping. Then for the next one, it's same 16. Then we refer to genetic paternity. If the mother is the one that is in doubt, we refer to that as maternity. For each, we've had each of these instances happening 50%. And in this case, half of what the genetic and so for relationship testing, the other half comes from and so for these two alleles that are seen for each of the loci, how one of them should come from the father and the other one should come from the mother. And so that is how the comparison is done for relationship testing. And so for here, I've highlighted the alleles that are similar between the father and the son. And if you look at these, this table, you can see that for each of the 24 markers that we've used, for each of them, there is a match between the deceased and then the alleged son. For each and every single one of them, there's a match between the two. A similar thing happened with the case number 3. Here, it's the father that is alleged. The deceased person is determined to be the son of this man. And so similarly, you're looking at the bolding ones being the alleles that are present for each of these two samples that were tested. On my discussions for case 1, Exhibit A, as I've indicated earlier, the main source of Exhibit A, or the only source of Exhibit A, is from the victim. But then Exhibit B is a mixture of profiles. And with that, the victim is the major contributor because it was an underwear of the victim. And so majority of the cells will come from the victim. But then minority, the suspect was a minor contributor. And so minority of the cells would come from the suspect. And in this instance, per the statistical analysis that was done, the mixture profile, the likelihood ratio and the mixture profiles that were determined were 4.10 times 10 raised to the power 17 times more likely to occur under the scenario that the underwear is a mixture of DNA from the victim and the suspect, as opposed to the scenario that it originated from a mixture of the victim and an unrelated person, an unknown and unrelated person. And this result is given to a probability or a confidence level of 99.99%. Same with C. But then if you look at C, because the suspect is the only contributor to the best sheet in this instance, where we did the sampling and then the extraction was done, the likelihood ratio is greater for this one as compared to Exhibit B, because Exhibit B is a mixture, and so it reduces the likelihood ratio that is indicated. And so then it was 4.10. Here is 9.30 times 10 raised to the power 27. And so in this instance, it's a great, since the suspect is the only person to have contributed to this, the conclusion made here is made on the calculated frequency on the DNA profile being rarer in unrelated individuals for any given population. For case 2, these are how we reported our relationship testing figures. The combined paternity index was 1.05 times 10 to the power 8. In this instance, as concluding statement was that the alleged relationship is not excluded. And so in this instance, the deceased is the true biological father of the alleged son, as in the bones that were found at the behind the hospital were that of the father of the person, the son, or the person that was brought to our end as a reference sample. Similarly, for this, the deceased person is the biological son of the alleged father. And so these were the three cases I wanted to. Thank you. I hope I have not gone beyond my time.
Yes. Don't worry about the time. In fact, the talk very interesting. Thank you so much. Thank you so much, Ima. Now I think we all right. Okay. I think no one has any question. Yes.
There were some network challenges.
Oh, okay, network challenge.
I would like to ask a question.
Okay, Dr. Abaji, go ahead.
So we hear of DNA reports and we hear of challenges from people who contest cases. Like they either they say they reject or they doubt. So I would like to know what could be the possible sources of these doubts or that they encounter from their report.
Okay.
Doc, can I seek some clarification? Are you looking at this in the light of paternity testing cases or criminal cases?
And also the case that you don't want to talk so much about, like the triggers cases.
Okay. We can talk about it. Don't worry. We can talk about it. I didn't want to bring it in my presentation, but then we can talk about it. I was okay.
So with regards to paternity case, actually I wanted to also ask about the mitochondrial DNA analysis, whether it's also done in the lab as well.
Okay.
All right. So please you can help. And also the case that you don't want to talk so much about, like the triggers case.
Exactly.
Okay. We can talk first one. I would indicate this since you want us to talk about the Takoradi case, we'll talk about it. One, I was part of the team that went to the scene. It was more of a mop-up scene. The thing had happened a year prior to that, and the suspect was leading them in different directions. So at a point, when we came on board, we decided to go back to the main areas or the primary crime scenes, and then do a mop-up of those areas and see what we can also get from that place, and then what the scene will talk to us about. So we went to Takoradi. Apparently there were two sites. That is one. The first one had to do with where the suspect was living. And so that was the first place we went to, because ideally that was the crime scene or the place we knew of. So we went there, we combed through the area, we retrieved some exhibits. Hello?
Yes, I'm here.
There's a bit of noise.
Can you mute? Your microphone, please.
Okay.
It's giving feedback.
I think it's better now.
Yeah, it's better.
We went to the first scene and then retrieved some exhibits from the suspect's room. Then we have still not found these places where the girls were. So based on a prior case that had been handled, we decided to check the manhole that was available in the house. It was sealed. And so we decided to check it and then see if we can, whatever we can get from that. So we opened it up and then we managed to retrieve, or we saw some skeletal remains in there. So we managed to retrieve those skeletal remains, wash them, collect them, package them, and all that. Now, before we left, apparently when the suspect broke out from jail, he had gone into a hideout. So our thought was, okay, let's just pass by the hideout and then see what we can also find there. So we went to the supposed hideout. We did a combing of the place. Initially we found nothing. Then when we were about leaving, we decided there were 2 wells there. So we decided to check these 2 wells. We checked the first one, there was nothing in there. We opened the second one. It was late in the night. We threw torchlights in there and then we saw these maggots come up in the water. And so it gave us some kind of delta to the place. So we decided that, no, we'll come back in the morning and then deseal or take out all the water from the well and then be able to see what we can get from that, why we are having these maggots in there. And then when we came back in the morning and did that work, we were able to retrieve one set of skeletal remains from. Later on, when we took those when we did some work with the pathologist, he was able to align or to determine 4 sets of skeletal remains. Now, here's the thing. You hear in the media of 3 girls in the Takrady case, but there were actually 4. For the 4th one, because the family did not want the thing to come out, they couldn't bring it out. As in, when the girl got missing and all, there was ransom that was paid by the family and all that. So they didn't want that one done. And more so, at the point we went in, that family in particular had more of accepted that at this point in time, their child was no more. So they were just seeking for some kind of confirmation. And apparently that was the 4th bone, the 4th skeletal remains we found in the well. Their house was a bit closer to that particular well. And so when these ones were picked, then later on, reference samples were picked from the family members. Profiles were generated and then comparisons were done. Now, to ensure that we had done everything right, all the procedure we did in the lab, we had to do it twice in a repetitive format. After we've done everything, we had to do it again just to confirm that everything that we've come out with was appropriate. The main challenge with that particular incident, I think, was issue of communication, which I don't want to go into that because that is not within my field. My field dealt with the exhibits that were recovered, the extractions, the DNA analysis process, and then the reporting that was done. And so I wouldn't focus or I wouldn't speak more on the communication aspect. because it was a very high-profile case. And then, obviously, some communications that was leading that whole charge as against the institution that I work for. And so that was probably where the whole gap was. But there was no problem with the exhibits that were recovered, and there were no problem with the profiles that were generated. The suspect, when we were retrieving the samples, the exhibits, especially the one at the well, the suspect was brought in to the site while the process was ongoing. And I'm speaking about it because the case has been brought to a close and the suspect has been convicted. The two suspects have been convicted. But then everything is done now. As I said earlier, it was a communication thing. And it's not my field, so I wouldn't speak much about that communication part. I hope I answered the first question.
Yeah.
Okay. And then the second question had to do with the mitochondrial DNA. Mitochondrial DNA is not a problem. I believe you know what it's for. It's mainly for, or ideally for, genealogy testing or for lineage testing.
Okay.
One, for majority of the work that we are doing, we are focusing mainly on forensic cases and then relationship testing. Relationship testing, we don't go as far back as 14 generations or 10 generations before you or so. It's mainly within the circles.
Okay.
The same equipment we have are the same thing you would use for mitochondrial DNA analysis. The only thing that will change is the kit. As I showed earlier, for extraction, for instance, there's a process for mitochondrial DNA analysis. And that is mainly indicated. There are several extraction procedures that you can use, but there's a kit for each process or for each sample you want to work with, depending on what you want to do. There's a kit available for that. And you can, you just purchase that kit, you do the extraction, go through the same process, and then you'll be able to come out with the results that you need. But then because we don't, for our line of work, we don't really focus on genealogy testing. We don't really apply mitochondrial DNA analysis. But it's the same setup, same everything within the lab that you'll be using for that. It's just a kit that will change.
Okay. All right. Doc, thank you so much.
Yo, thank you very much.
Dr. do you want to follow up?
I'm cool for now.
All right. Dr. Edgar. Dr. Edgar, you've raised your hand.
Hello, yes, good afternoon.
Good afternoon. Yeah.
Hello, Super, thank you very much for your insightful presentation. For those of us who are not scientists, we were only dangling through the loci and things like that. But I think the whole thing, the whole essence of forensic profiling is to help the criminal justice process.
Yeah.
My concern, we have a couple of criminal justice students online. My concern is, how does the whole process facilitate the work of prosecution? Again, do you have
Hello?
Hello? Dr. Edgar? Dr. Edgar? Are you still there?
correct?
Okay. Can you come again? I think we didn't hear the second aspect.
Can you?
Yeah,
Can
we
you hear
didn't
me?
hear the...
No, okay, I'm on it. So I'm saying that the whole process of forensic profiling or investigation is to help the criminal justice process in criminal cases. I want to find out. I want to find out if somebody, you have had cases where people contest your results, and if it happens, what are the options available for redress for such a person? And again, I'm just putting myself in the shoes of the judges who may not be scientists like me. Does it mean that once you bring this thing to court and say that we have evidence to support it, that is the end?
Okay.
Please, did you get my question?
Okay. So yeah. I think it's 3 questions, so it's 2.
It's almost 2, but yeah, 3 questions, yes, please.
Okay. 3 questions, yeah.
Okay. So let's look at it this way. I think for the first one, I would look at the forensic aspect and then the criminal aspect, the relationship testing aspect. I'm dealing with the in helping the I think the first one had to do with helping the criminal justice system.
Yeah, hello? I think the first, yes, it's true. It's how the forensic analysis helps in the criminal investigation process.
Okay. I think maybe I should have indicated earlier that the major role the lab plays or the forensic process plays is to support the criminal justice process. Now, let me sound this. Because it's a forensic lab does not mean the lab is there to convict people. The forensic process either convicts or exonerates. It is the application of science to the criminal justice process. And this science can come in several forms. Today, I didn't talk about chemistry. I didn't talk about the narcotic cases, the document examination, and all the other sections we deal with. I focused on the DNA analysis today. And yes, I accept it was a bit technical today because I had to go deeper into the entire process that we use in coming out with a result. And so there was going to be majority of it being technical, and more so because I was dealing with some forensic science students. I think a bit of it centered on them, and that is why the interaction stage is here now, so that especially for those who are not in that area, it will be clearer to them, and then you'll be able to get it. Back to the question. When we do our work, most of the times, especially with the forensic cases, most of the times the case is already in court. So we do our work. We submit the report to the court. As I mentioned earlier, DNA analysis is the gold standard globally. But then, as it's indicated in the law, I think Dr. Kwachi Nwaku can bear me out that the justice usually lies in the bosom of the judge, or the conclusion of the matter usually lies within the bosom of the judge that sits to adjudicate the matter. The prosecution will present its case. We come in as witnesses or expert witnesses to bring to bear what the science of the scene is speaking, or what has been proven. Now, there are instances, and even this one is not just here, globally. There are instances you present all this, and the judge will still say no. Okay. But then for majority of the times, when this comes out, especially for the judges that now do understand, when this report comes out, that is more like a concluding matter for them. When it comes out, or when we send our reports there, that is more like a concluding matter for them. To the other question, where you asked about seeking redress and all that. If it's a paternity case or a relationship testing case, for us, we do the relationship testing case to support the work that we do, because it's the same reagents we are going to use anyway. If it's a relationship testing case, if we do it, usually when you come, we take your samples, we pick it in duplicates. And so we use one for the work that we need to do. Then we reserve one. Should, in case you want a redress and you want to take that same sample along to a different place, we'll provide that same sample to you so that you can use that to do your relationship testing. If it's relationship testing, you can do it, the redress, you can do the redress probably at a private place. But if it's a forensic case, as in a criminal case related, obviously it's only a forensic lab that can handle it. And so in that sense, it would have to be referred outside. And it would have to be referred to a forensic lab outside of your choosing. We can give you the various ones available, and then you would choose, and then it'll be taken there. It's taken there.
Okay.
Yeah.
All right.
Thank you very much. Yes.
All right.
That's good. Thank you.
You're welcome. Yeah. Okay. I think Amadou, you've raised your hand. You can go ahead to ask your question.
Yes, sir. Good afternoon. So my question is whether the Ghana police are making efforts to extend their forensic labs to other regions, because this is the case where the Ghana police only have their lab in Accra.
Oh boy. Okay. My boss is here.
Yes, your boss should answer this for us.
Anyway, but just to give a little brief, maintaining a forensic lab is very cost intensive.
Yeah, very expensive.
Globally, it's very cost intensive. Even the UK, at a point, and then currently now, they have privatized their forensic lab because the state was no longer able to fund it. And so they had to privatize the forensic lab. So they will take their samples there and then pay for it as and when the samples come in. And so it is very cost intensive. Even with the history that I gave earlier, I indicated in 2010 that there was some renovation and then the DNA section came on board and all that. That was through a grant by the European Union to the government of Ghana and the Ghana Police Service. Okay. And it was a huge sum. Now, I can tell you for a fact, let me show you the page that I showed some of. If you are looking, I believe you can all see this.
Yeah, we can see that. Yes.
This small box here alone on the down right corner.
Yeah.
The small box there alone cost almost $10,000.
Wow.
And it has an expiry of 6 months. You have to order from abroad. By the time it gets here, almost 3 months are gone. So you have just 3 months to work with. Okay. So it's a huge challenge. I'm not holding brief for anybody, but those are the realities on the ground. Since 2010, we've proposed to our administration and then also to the government of Ghana to extend the labs to the various regions, if not all the regions, at least for Ashanti and then maybe Northern, to cater for the various belts within the country.
Yeah.
Because it's a challenge having to probably in Ashanti, and maybe another one in the Northern, or if we can't even do the Northern, at least another one in Ashanti to cater for the Northern belt. Obviously, it would lessen the work on us. For our units, there are 6 analysts and 2 technicians or 2 assistants helping out. Currently, one of the analysts is out, and it's most likely not coming back to the lab. The other one has to do with is also out in the US. I don't know if he will come.
Right. I think you've given us a lot of insight.
But my boss is available. Probably he might want to chip in something.
This COP. Yeah. Hello.
Or maybe he's off.
I think he's off. Yeah.
Oh, okay.
Anyway, yeah, I think it's good. Ima, I think it's true the question the gentleman asked, but I also want to ask. Before we had forensic science in Ghana, how were you getting your human resource from?
Is it in terms of the DNA lab or the entire
DNA lab specifically?
Okay. So the simple thing that was being done, I mentioned earlier that it was just serological analysis that were being done.
Yeah.
And so you do tests to identify whether it's semen, human semen, and then you do blood grouping. Those were the main things that were being done then.
Okay.
If it was a high-profile case, then they would take the sample out, mainly to South Africa, for DNA analysis and then pay for it.
Okay.
Mm-hmm. Until the lab was set, the DNA section was brought on board. So prior to 2010, you'll be hearing of if it's a high-profile case, let's say the Atai case and all those other cases, you hear that samples were being taken outside to South Africa.
Okay.
That was what was being done during the process during those times. And then for paternity testing too, it was still being taken outside. Currently, even for paternity testing, majority of the private labs will take the sample here and then take it outside for analysis. Yeah.
Simply because the analysis is very expensive.
1, very expensive. 2, the experts to handle it for them will not be available here.
Okay.
So the most cost-effective one will be to take the samples and then ship it out to South Africa or to US or whichever place that the private company has a relationship with. Take it there, have it analyzed, it's brought back, and then they issue reports out, paternity reports and all that to people that this is the outcome.
Oh, I see.
Yeah, so that is what
wonderful. Okay.
Kolebu started when we came on board. Kolebu, I think 2 or 3 years later, came on board, and then they ran for I think about 3 years, and they could not. So all the samples that they had accumulated during that period, they had to send it to our end.